Did you know not all microbiology labs are the same? Some are safe enough for students, while others handle viruses so dangerous, even a tiny mistake can be deadly. These labs are divided into 4 levels, BSL-1 to BSL-4. Let's break them down, from safest to most dangerous. Biosafety Level 1 is the most basic level of laboratory safety. It is used for microorganisms that are not known to cause disease in healthy humans. These labs are commonly found in schools, colleges, and beginner research labs. Standard safety practices are enough, like wearing lab coats, washing hands, and keeping the workspace clean. There's no special containment, because the risk is minimal. But not all microbes are this harmless. Biosafety Level 2 handles microorganisms that can cause disease but are usually not severe and can be treated. Here, safety rules become stricter. Gloves and lab coats are mandatory. Work is done inside biosafety cabinets. Lab access is limited. These are pathogens you might encounter in everyday life, through food, water, or surfaces. But what if the danger is not just contact, but the air itself? Biosafety Level 3 is where labs become high-security environments. These labs deal with pathogens that can spread through the air and cause serious or even fatal diseases. Now the lab itself becomes a containment system, controlled airflow, negative pressure, specialized ventilation systems, researchers wear respirators, strict access control. In these labs, even the air is controlled. And then comes the final level.
If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated.
Biosafety Level 4 is the highest level of biological safety. These labs handle the most dangerous pathogens known to humans. Viruses that are often untreatable, highly infectious, and sometimes fatal. Everything here is designed to prevent exposure. Full-body air-supplied suits, airlocks and sealed entry systems, chemical decontamination showers, isolated air and waste systems. This isn't just a lab, it's the last line of defense. Labs to extreme containment facilities, each level exists to protect scientists and the world. We'll break down the design of these labs and what makes them so secure. Ever wondered what makes these high-containment labs so secure? It's not just protective suits, it's the entire design of the facility. Biosafety laboratories are built using a layered design system. Each level adds another barrier between the pathogen and the outside world. From basic Biosafety Level 1 labs to advanced Biosafety Level 4 facilities, the goal is simple: containment, control, and protection. Access to these labs is strictly controlled. You don't just walk in. High-containment labs use multiple entry barriers, including security checkpoints, changing rooms, and airlock systems. Each door must fully close before the next one opens, ensuring nothing escapes unintentionally. One of the most critical safety features is airflow control. Air always flows from clean areas into contaminated areas. This is called negative pressure. Negative pressure ensures that even if there's a leak, air moves inward, not outward, keeping pathogens contained. Inside the lab, work is never done in open space. Procedures are performed in biosafety cabinets, sealed, ventilated workstations that protect both the scientist and the environment. These cabinets act as an additional containment layer during experiments. Before exiting, everything must be decontaminated. Personnel pass through chemical showers, and all materials are sterilized using high-pressure steam systems. Autoclaving ensures that no bacteria, virus, or contaminant leaves the facility. Before we move into waste management and filtration systems, don't forget to like, subscribe, and share for more in-depth microbiology insights. Nothing leaves the lab untreated. Air is filtered through HEPA filtration systems, and liquid waste is chemically treated before disposal. This ensures complete environmental safety. In the highest containment labs like BSL-4, scientists wear full-body, air-supplied suits. These suits are not just protective gear, they are part of the lab's integrated safety system. Every wall, every door, every airflow system is designed with one purpose: to prevent exposure and protect both scientists and the world. You've seen the layers, now here's the real difference. At Biosafety Level 1, safety depends on the person. At Biosafety Level 2, it shifts to equipment. At Biosafety Level 3, the lab itself becomes the barrier. And at Biosafety Level 4, everything is containment. The higher the level, the less you rely on people, and the more you rely on systems. That's what truly separates each biosafety level. But what actually lives inside these labs? Let's look at real-world pathogens, level by level. At Biosafety Level 1, scientists work with organisms that are generally harmless, like Escherichia coli K12, a safe strain used for teaching and basic research. These microbes don't pose a threat to healthy individuals. At Biosafety Level 2, the risk becomes real. Pathogens like Salmonella enterica, Staphylococcus aureus, and viruses like hepatitis B virus are handled here. They can cause disease but are typically manageable with proper precautions. At Biosafety Level 3, we deal with serious airborne threats, like Mycobacterium tuberculosis and SARS-CoV-2. These pathogens can spread through the air, making containment absolutely critical. And at Biosafety Level 4, the most dangerous pathogens on Earth. Viruses like Ebola virus and Marburg virus, highly infectious, often fatal, and with limited or no treatment. These aren't just microorganisms, they are the reason these labs exist. Each biosafety level isn't just a precaution, it's a response to the threat inside. Curious where each pathogen belongs? Global agencies like the World Health Organization and Centers for Disease Control and Prevention publish official biosafety classifications used by labs around the world. So how does all of this work in real life on a daily basis inside the lab? Before anyone even enters a biosafety lab, they must go through strict training. This includes understanding protocols, emergency procedures, and how to properly handle infectious materials, because even in high-tech labs, human error is still the biggest risk. Every step inside the lab follows a defined protocol, known as Standard Operating Procedures, or SOPs. From how to hold a pipette to how to dispose of a sample, nothing is left to guesswork. Work surfaces are disinfected before and after every procedure. Common disinfectants like ethanol and sodium hypochlorite are used to eliminate contamination instantly. If something goes wrong, like a spill or accidental exposure, there are immediate response protocols. Areas are isolated, disinfected, and incidents are reported and documented. Lab waste isn't just thrown away, it's carefully separated into categories: infectious, chemical, and general waste, each with its own disposal method. Labs are regularly inspected and monitored to ensure biosafety standards are maintained. This includes airflow checks, equipment validation, and protocol compliance. So biosafety isn't just about advanced labs, it's about discipline, consistency, and strict procedures. Because in the end, even the most advanced containment systems depend on how well they're used.
Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!